in this context. Okay, so we know that we're going to have some tubes. If we need something to move something through the tubes, what are we going to expect to find in those tubes? Those tubes have to also be made of what? Muscle. Okay, so all of the organs along the digestive tract are going to be filled with muscle. Now, they're also going to be hollow. Why are they hollow tubes? Well, so that we can put something into the tube to move it through it, just like a piece of PVC pipe is hollow. Why? So that we can move something through the PVC pipe. Okay. So in this case, we have a PVC pipe that is made out of muscle, so that that muscle can squeeze, just like toothpaste, and squeeze the toothpaste out of the tube. Okay. Now, what are we also going to see on the insides of these muscular tubes? So we know that, say we take that bite of chicken finger. Are chicken fingers smooth? No. So they're kind of rough, right? So if you swallow one, it's going to kind of scratch a little bit too, right? So we also have to now be able to deal with that abrasion. So what are some tissue types that you can think of that might help us deal with that abrasion? Do we want thicker tissues or thinner tissues? Thicker. The thicker something is, the better it's going to be able to deal with scratching and scraping, right? Because you can hurt it a little bit and there will still be some of it left. Does that make sense? Exactly. When it's thinner, you could easily scratch it. It could tear. We use thin stuff if we just want to move stuff across it really, really, really fast, right? But we use thick things like on the palms of our skin, just because, or the palms of our hands, just because these are constantly touching stuff. So we need that as protection. So we're going to have thicker tissues where we're expecting lots of abrasion, like say in your mouth and your throat, because sometimes you don't chew your chicken finger enough and you swallow it and it scratches. Okay, so we need protection from that. So we're going to expect to see epithelial tissues lining all of these things. We're going to have thicker ones in places that are going to be dealing with lots of abrasion. We're going to have thinner ones in places where we need lots of absorption. Okay, we're also going to have a lot of cuboidal ones whenever we need to be dealing with our secretions, because remember, cuboidal cells are the things that typically secrete. Okay, so that's going to be some of the stuff that we're expecting to see. Now, let's go ahead and look at a general layout of the organs, and let's think about what we're going to be kind of doing. We're going to go through each individual one and kind of talk about their general function here. But let's think about or look at kind of the overall layout of the system though. We start with our mouth and we enter our esophagus, a muscular tube. We enter our stomach, a continuation of that same muscular tube. Let's follow it down. We leave the stomach and we enter the small intestines, which if we look at it up on the screen, looks like what? A muscular tube. Small intestine. Wait, large intestine. Wait, still a muscular tube. And then out. This is all nothing but a muscular tube that is open to the outside environment on both ends. It is basically the outside. Okay. It is a big, convoluted muscular tube. One way in, one way out. Okay. Now, those tubes are what we actually call the GI tract, or the gastrointestinal tract. Okay. If you've ever heard someone say a GI infection, it is an infection of specifically some part of this tube. Okay. Now, associated with this tube, we have a bunch of accessory structures. The accessory structures are going to help us do the jobs that we need the tube to be doing. Because remember that in the tube, all we're really doing is moving things from point A to point B. And this is inside the tube. We are going to chemically digest these things. We are going to absorb these things. That's really all we can do in the tubes. Anything else that we need to have happen is going to have to be helped by some of these other organs. Okay, so we need to break our food down. How do we break our food down? We break our food down with teeth. We need to make sure that the food we're eating is not going to kill us. So we need to taste it. Okay. We also need to lubricate its ability to move down our esophagus. So we need saliva. So we have our salivary glands. That saliva is going to help that food slide down and not like just tear its way through that little muscular tube. Then once it gets into our stomach, we're going to squash it all up with a whole bunch of hydrochloric acid, start to break down some proteins. And Once we've started to break it down a little bit, we're going to move it into the small intestine. But on its path, or once it gets into the small intestine, your gallbladder and pancreas and your liver are going to secrete some enzymes into that liquid to make sure that we can finally finish breaking down that food. Okay. Then it gets into our small intestines. This is where we finish digesting. This is where we absorb all of those nutrients. Your large intestines is where we're going to absorb the water from it. And then finally, we are going to expel the rest as waste. Okay. All right. So here we have the tube. Okay. Now, different parts. So whether it's the esophagus or the stomach or the small intestine, large intestine, they're all going to vary a little bit, but they're all going to have this same general structure. Okay. We're going to have an opening that we're going to call the lumen. That lumen is going to have on its inside, we're going to have a layer called the mucosa, which is going to start with an epithelial tissue, a lamina propria, which is the connective tissue, and then we're going to have a very small band of muscle under that called the muscularis mucosae. Okay, muscularis mucosae. So it's a layer of muscle in the mucosa. Mucosa also tells us something important. What are we secreting here? Mucosa. Mucus. We have to have mucus. The mucus provides protection in the form of lymphatic associated lymph tissue, or like mucus associated lymph tissue, holds on to certain things, but it's also going to act as a lubricant in a lot of ways to make sure that the stuff can move through. It's also going to protect our epithelial tissues from the ridiculously low pHs that we're going to find both in our stomach and in our small intestine and even in your large intestine. Okay. For instance, or to put this into perspective, the reason that kids need diaper cream and stuff like that is because feces is incredibly acidic. We neutralize it as much as we can in our intestines so that it does not just kind of eat its way through the intestines. But your stomach acid is so strong that a bunch of that acid is still there, even in the form of feces. So if you leave a bunch of feces sitting on the surface of the skin for a little while, it tends to eat away at that surface and it gets incredibly irritated very, very, very quickly. Okay. That's what diaper creams are there for. That's also one of the reasons that if you look at the ingredients on diaper creams, the first one on almost all of them is going to be some sort of basic chemical, a base so that we can neutralize the acid so we can stop the pain, okay? Or at least stop the damage. The rest of it will typically be some sort of local anesthetic, okay? Now, we have to have mucus here then. Now, a bunch of these tubes are also going to have a whole bunch of these little convolutions here. So if we see all these little fingers, and we see all these little kind of circles in here, this is going to give us a whole lot of stretchability. Anytime we need something to stretch, what we do is we basically build the tissue as though it is much, much larger than it is, and then shove it into a small bag. Okay, so just like if you take a balloon and it's loose, it's very, very kind of tight, right? It's kind of constricted on itself. But if you take one and you blow it up really, really big, or you stretch something out a whole lot, so say you stretch your shirt really far, and then you let it go, it kind of crinkles back up, right? So those crinkles tell you that you can go larger than you started with, okay? So these little crinkles basically tell us that we can stretch this out a little bit. Now, under that mucosa, we're going to find a layer called the submucosa, and then we run into the muscularis, okay? So the muscularis, well, muscle, okay? So we're gonna have a couple of layers of muscle here. We're gonna have a layer of muscle that's going to run longitudinally. That's going to be this outer layer here. And then we're going to have a layer of muscle that is going to be circular. Okay, so when the circular one constricts, it makes the opening smaller. When the longitudinal one constricts, it opens it up. So now, all of a sudden, if we constrict on one end and we open up in front of it, when that muscle constricts, it's going to be able to push something into the region that opened on the longitudinal layer. Okay, and then finally, we have the serous layer on the outside. Okay. Now, also associated with this, or with these tissues, especially in the intestines where we need to do absorption, we're also going to find capillaries very, very close to that epithelial lining, is both blood capillaries and lymphatic capillaries. The reason these lymphatic capillaries are important is because a bunch of the stuff that we're going to eat, so say you eat that chicken finger. Well, that chicken had fat in it, and there was also a bunch of fat that got trapped in that breading. So, is fat water soluble? No. So if fat is not water soluble, is it going to be very easy to absorb that stuff into the blood and move it through the body with the blood? No, not particularly. So what do we do then? Well, we absorb it using our lymphatic capillaries. Okay, so we absorb it into the lymphatic capillaries so that we can move it around and then dilute it into the blood or connect it to some sort of chaperone protein so that we can then move it to the blood that way. Okay. So when those longitudinal muscles contract, they open the opening of the GI tract. So the longitudinal muscles contract and it kind of makes the lumen larger. And then we will constrict the circular ones, and that will allow us to push a bolus of food like this through the tube. Okay, that's what you're actually feeling happen when you kind of swallow that too big bite of food and it kind of feels like it's stretching its way down. You are feeling it stretch the muscles. Okay, so you're going to get that sensation from those proprioceptors in the muscle, but you're also feeling those muscles constrict behind it, helping it push its way down. Gravity is not strong enough at that point, so we have to push it down. So that's part of what you're feeling in those circumstances. Okay, now we can also take those foods though, and it's basically like taking 2 baggies of food or 2 cups of water and mixing the 2 things together. We kind of just push them together and allow them to mix that way. And then once everything's mixed up, then we can peristalsis its way out. When you feel your stomach growling, what you're actually feeling is your stomach trying to do this bit. It's trying to mix up the contents of your stomach. The problem is that you haven't eaten anything, so there's nothing for it to grab onto or nothing for it to mix. So all you feel is it pushing that air out. That's also usually why whenever your stomach is growling, you tend to be a little bit gassy. All right, so now let's go ahead and talk about some of the, we've already talked about that a little bit. All right, so now let's go ahead and work our way down through the tube. Okay, we know that we of course start with our mouth. Now, in your mouth, we of course have teeth, okay? So your teeth are here to do what to our food? To break it down. So your teeth are there to help us break down the food, but to what end? All right, so say we eat a Snickers bar. Snickers bars are typically square in cross section, right? So here we have a Snickers bar. So what is the outer layer of a Snickers bar typically made out of? What? Chocolate. All right, so here we have the chocolate. What else do we find in a Snickers bar? I don't know why I'm quizzing you over the contents of candy. We have peanuts. Caramel. And whatever nougat is. And whatever nougat is the best part of this, but hey, to each their own. Actually, I guess the caramel's caramel. Then nougat. Seem relatively close for what's in a Snickers bar? Have I actually bitten to one to look at one in cross section? Yes, I have. Okay, I'm a nerd. I'm also a little strange even then. All right, so we have a Snickers bar. Say you take one of those bite-sized Snickers bars, you pop it in your mouth and you just swallow it. Not saying you're going to choke, but what is the only thing that you're going to feel or taste? The chocolate. The chocolate is the only thing that's at the surface. Okay? So if our goal is to break this stuff down into pieces, what we've just done is we haven't done that. Only the surface area of the chocolate are we going to be able to do anything with. Now, each of these is made out of different things. The chocolate is made out of something different than what's in peanuts. Caramel has different ingredients than chocolate. Nougat has different ingredients than all of them too. Okay, so our goal is to turn that chocolate into the individual chemical components that make it up. So chocolate is composed of cacao, which is basically like a little bitter protein thing, or like a bitter substance. Okay, so it's like a little bitter molecule. It's also composed of milk fat and sugar. So we were trying to turn it into fat, and sugar. Individual sugars, okay, and individual fatty acids. Caramel is composed of sugars and fatty acids. Peanuts are composed of just fatty acids, basically. Nougat is really just sugar. I don't think there's anything else to nougat. I think that's all it is, okay? Maybe some tiny little bits of protein to hold it all together, okay? Now, we're gonna do that using chemicals. So what do we call the chemicals that catalyze reactions? The chemicals that do things in our body. What do we call those? Those are the enzymes. So enzymes, are enzymes larger or smaller than a Snickers bar? Smaller by a lot, right? So an enzyme is not going to be able to eat the entire Snickers bar. The enzymes only get access to the surface. Okay, so if you do not chew your food, you do not have, or you do not give your enzymes access to the surface of the food. Thus, you can't taste the peanuts. You cannot taste the caramel, nor can you get anything out of it. You don't believe me if this is how it works. I dare you to go home and just swallow a baby carrot whole. It's possible, you can do it, okay? Just be careful, you don't actually have to do it, because if you do and you die, I don't want to get blamed, okay? But you can do it. Then watch what happens the next day when it comes out. Or say you eat a piece of corn that you don't chew, because usually when we're eating corn, we don't get a chance to bite all the pieces in half. So what happens in a couple days? What do you see? Corn, whole and complete, and completely undigested. So is there any point to you eating that piece of corn? Did you get anything out of it? Nope. Surface area matters. If we don't chew it, we can't break it down. So what we're doing, every time we cut this thing, what we're doing is we're more than doubling the amount of surface area that we have access to. The more surface area that we can create for our enzymes, the better breakdown we can make. So our goal in the first part of the digestive system is exactly that. It's to make as much surface area as possible. So in our teeth, what we're trying to do is we're trying to start what we call mechanical breakdown. We call it mechanical breakdown because we are breaking it into physical components, large components. The chemical breakdown will come later. However, we do have an enzyme in our mouth that will already do this. In our mouth, we produce amylase. So from our salivary glands, we make amylase. So amyl, ASE. ASE tells us it's an enzyme, right? Amyl then is the root. So what is it breaking down? Amyl is actually old Latin root for sugar. So it breaks down sugars. If you put a piece of bread on your tongue for a couple seconds, it will start to taste sweet. The amylase in your saliva is going to start to break down the complex sugars in the starch into individual sugar molecules. So the bread will taste sweet. That's part of why we like to eat bread and potato chips and things like that, is because they're a great source of sugar, and our brains crave sugar because the only thing that your brain burns for energy is sugar. That's it. So anything that has sugar in it, your body is just automatically like, hey, that is fantastic. That's actually literally why you can see the color red, so that we could recognize ripe fruit. Okay, so salivary glands are going to help us break down some of these complex sugars. But do they get access to those complex sugars in, say, the nougat or the caramel if we don't chew the food? No. So we would not be able to taste the lovely caramel on the inside of our Snickers bar if we don't break it. Okay, so your teeth start mechanical breakdown. Okay, so instead of being left with all these things, we're going to get now pieces of chocolate. I need a different blue marker. Is there one? There is not. Oh well, we'll survive. It's going to start the breakdown of the chocolate. So we're not going to have pieces of chocolate. We will have pieces of nougat and pieces of peanut. Okay, So we're going to get pieces of everything. Now we can taste all of these things. So we start the process of mechanical breakdown. Now, we have taste receptors, of course, found in our tongue. Those taste receptors, remember, are found at the bottoms or at least at the sides of those papillae, which means in order for us to taste the food, it also has to get dissolved a little bit. Once again, if you don't chew your food, you don't taste it as well. If you've ever noticed when people are like doing a taste test on a show, they take small bites, but then they chew it a bunch. They let it stay in their mouth a while. Why? So that more of it gets dissolved so they can taste it better. Okay. So we need to, once again, chew our food just so we can get more small pieces so that we can taste things. But that's in our teeth. We still have them as noticeable pieces of peanut, noticeable pieces of caramel, noticeable pieces of chocolate, and things like that. Okay. Now, your tongue is a giant muscle. Okay. If you look at it here on the screen, you can see that your tongue is basically this big fan-shaped muscle. Like you can press your tongue against the top of your mouth, right? You can press it against your hard palate, which is the bony part towards the front, or the soft palate, which is the softer part behind that. And your soft palate extends down here to the uvula. But you can actually take your tongue and press it all the way up against the front of your mouth, at which point you can push your food towards the back of your throat. When you go to swallow, that's actually exactly what's happening. When you go to swallow, your tongue presses up against the top of your mouth and forces the food towards the back into your pharynx. Okay. Now remember, your pharynx has 3 spots. We have naso, oro, and laryngopharynx down here. So it's going to push your food towards the esophagus. At this point, we also need to keep the food from going into our trachea. So this is where your trash can lid closes. This is part of why your mom told you to close your mouth while you were chewing. It's because if you have your mouth open while you're chewing, you are breathing. So it's very easy to both swallow and take a breath in at the same time. When you do that, that piece of rice you just ate will end up down here, and you will cough and hack until you blow it up out through your nose. I say this from experience. Okay. This is also why you can accidentally sneeze stuff out through your nose, like when you're coughing and hacking and stuff like that. It's because it gets pushed out of your trachea and up through that nasopharynx. Okay. So we're going to push it back here towards the esophagus. Notice that the esophagus is ordinarily closed. This is a tube that is made out of muscles. So those muscles are kind of constricting it and making it fairly small. It will open up only when we swallow. So when you swallow, your mouth or your tongue presses up, and then we're also going to relax the muscles back here at the back of our throat to allow the food to slide back. Okay. Now, to help us do this, we also have, is there a slide? Okay, there we go. We also have our salivary glands. Now, your salivary glands are 3 sets of tissues. There are 3 glands in 3 different places. We have one found underneath the tongue, so sublingual, under lingual tongue. So sublingual salivary gland is going to live under your tongue. It's got a whole bunch of small ducts, which are going to allow it to pass its liquids through. Okay. Then we have our submandibular gland. What do you think this one's going to be under? Mandibular. The mandible. Okay, so this one's going to be underneath your jaw. Okay, so your submandibular gland is going to have a duct that is going to release right here behind your front teeth on your bottom jaw. You can actually see this. So if you actually wanted to look in a mirror and kind of stick your tongue up towards the top of your mouth, or you can just take a picture with you lifting your tongue, you'll be able to see 2 little tiny holes just behind your front teeth. Okay, those 2 little tiny holes are actually the outputs for your submandibular glands. Okay, now we also have the largest one that produces the most saliva back here, and this is your parotid gland. Now, your parotid gland is going to sit on the surface of your masseter. So the masseter muscle is that big muscle that lets us chew. Okay, so if you kind of clench your jaw, that muscle is the masseter. The big soft spot that connects basically the front of your ears to that masseter, that is all your parotid salivary gland. This is also what gets infected when someone gets the mumps. Okay, so when someone gets the mumps, the infection kind of starts here, and you'll actually see them develop the mumps, which are these big swellings of the submandibular gland. So it looks like they'll get these kind of chipmunk cheeks. okay? And that's because of the infection of the parotid glands. Now, on the insides of these glands, you're going to see a bunch of these. Okay, so these are basically going to be, well, an alveolus. So it's going to be like those hollow grapes, like we looked at in the lungs. But in this case, they're lined with cuboidal cells that are secreting saliva. Okay, so we can see these little grape-like clusters. And your glands are basically just hundreds of these grape-like clusters. Okay. Now, oh, I guess I went past it. What we're looking at in saliva, though, is mostly going to be water. Okay, it's something like 99.5% water. And we make around a liter of it a day, anywhere from 1 to 1.5 liters a day. This is 1 liter of water in my hand. This is how much saliva you swallow on a daily basis. Okay, one of the options for labs to make this lab more fun, talking about digestion, is for us to do a saliva lab. And I'm not going to do it because it is nasty. Okay, it is gross and it smells awful. Like, no, we're not doing that. Okay, I don't care if it is the best learning experience ever. We're not doing it because it's gross. Okay, and I would throw up, so I don't feel like doing that. Now, this is how much we make, but the vast majority of it we swallow. Okay, so you don't really even notice it's there. We swallow it because that's the point. Okay, it is there. It also has some enzymes in it, some mucins and lipase. So there's a lipase to help us taste fats, and there's also an amylase to help us break down sugars. Okay, so we can taste things that are sweet and we can taste things that are fatty, in part because of the enzymes in saliva, but most of it's water. But another thing that it's trying to do is to help us swallow our food. Okay, it is basically acting as a lubricant to make it easier for us to move the boluses of food through the digestive tract, or at least through the esophagus. Now, we call this process of chewing our food, we call that mastication, okay? We masticate with our teeth. Your teeth are also the hardest bones that you have in your entire body. The single hardest substance that your body can produce is this, enamel. Enamel is what's going to be found at the surfaces of your teeth. It is this incredibly, incredibly hard, protein-rich and kind of mineral-rich substance. It is incredibly, incredibly, incredibly tough. So tough that, like, you can bite through fishing line fairly easily with your teeth. You should not, because it's a good way to click your teeth together and break your teeth, but you can do it. Okay, why do you think our teeth need to be so hard? To break down everything we eat. Think about some of the stuff that you put in your mouth. Like, they're Jolly Ranchers, Tootsie Roll Pops. Don't eat those because they'll cut your teeth. Or not cut your teeth, but cut your mouth. Beef jerky is tough. And those are just food items. People put pens and pencils, and you don't do that. It's gross. Put pens and pencils and fingernails and stuff like that in their mouth. Feel how hard your fingernails are. You can hear that. You can bite through that with your teeth. That is why why your teeth have to be so tough, is to be able to deal with all these kinds of forces. And we do it over and over and over again. That's a lot. That's a lot of force over the span of our lifetime. So it makes sense that the enamel has to be incredibly tough. Now, after you get through the enamel, we're going to get into this substance here. This is dentin. Dentin is also tough, but it is not quite as tough as the enamel. Now, on the insides of your teeth, because your teeth are hollow, you're going to find these massive networks found in what we call the pulp cavity. We're going to find these massive networks of nerves and blood vessels. So yes, your teeth can bleed. Okay, there are blood vessels in here. We need blood vessels in here so that we can make all the cells that live inside your teeth and make all this dentin and stuff happy, right? So we have to have blood vessels going to them. But they are connected into the bone. All of this stuff right here, this white and yellow, this is all bone. This is inside your jaw. The feet, or the roots of your teeth, are buried in the bone. Then they are basically welded to the bone with additional ligaments called denticulate ligaments. So not only are they sticking in the bone so you have more surface area, like of bone touching bone, you then tie them together with a whole bunch of little tiny ligaments. That is why it is so hard to remove a tooth. You don't believe me? You can break a tooth before it actually releases from the jaw. That is how tough these things are. Like, it is insane how tightly they are bound to the jaw. However, I also said that there is a nerve here. A nerve runs into each little foot. Okay, we call this little pathway right here going from this pulp cavity down through the roots of the teeth. We call that the root canal. That nerve is what is sending the signal out that says, hey, this effing hurts whenever you have a toothache or when someone breaks a tooth. That nerve gets very, very angry. If you've ever known someone to have a root canal, that's because they're trying to go in here and basically say, hey, nope, the nerve, you don't need to be here anymore. And they'll go in and kind of cut the nerve out basically because that tooth is wounded and injured. So they basically try and stop it from sending the signal out that says, hey, this is hurting. Okay. But that's also why it hurts like hell to remove a tooth, is because of these big connections down in here. Now, this is a molar. So molars have more connections. Your far back molars actually have 3 roots instead of 2. And as you move forward, they get a little fewer. Now, children, like we of course know that we started with what we call baby teeth or deciduous teeth. Deciduous just simply means that they fall or that they are replaced. Trees that drop their leaves are called deciduous trees. Okay, so deciduous teeth are the teeth that we will tend to lose. Now, we grow, like I said, 20 of these. As adults, though, we have 32. And this is true for basically all mammals, okay? Or at least all omnivores, I'll put it that way, okay? So we have 32 permanent teeth. These replace those deciduous teeth. Those deciduous teeth get pushed out basically as these permanent teeth are growing in. Most of your teeth are done coming in, or your teeth are done being replaced around your late teens, early 20s. That's when we get our 3rd molars, or what we call the wisdom teeth, okay? They're called wisdom teeth because they arise when you should have gained at least some wisdom. Like, hey, Matt, don't decide it's fun to jump off the roof of your house onto the ground. Okay. That is something that I learned. I had to gain some wisdom in the process. Okay. Now, we have 3 different types of teeth. Oh, now one thing, whenever you're looking at these pictures, these are the baby teeth. We know they're baby teeth because they're small. This also tells you when they arise. I want y'all to keep in mind that there's a hell of a lot of variation here. You are not weird if you did not get your last set of molars until you were 4. That's fine. That's actually not terribly abnormal. Okay. Even the orders that these things come in is not set in stone. There is a lot of variation here. My daughter, for instance, is like, I think 20 months old now, thereabouts. She just now had her bottom canines come in. Her top ones came in first, but she actually had premolars come in, and even her molars starting to erupt. First sets of molars starting to erupt before her canines even came in. So lots of variation here. Same with the adult teeth. There's lots of variation on when these things come in, but these are at least give you some kind of general rule of thumb for when these teeth come in. Now, we do have a few different types of teeth. We have incisors. These are the flat teeth at the front. These are good for shearing things. So if you ever wanted to kind of bite some fishing line, you typically, or bite your fingernails, you typically do that with your front teeth because they are really good at basically being like little scissors. Okay, so really good at kind of shearing stuff off. Okay, then we move back and we have the canines. The canines are good for starting to tear things. Okay, so your canines and your premolars are what we tend to use whenever we need to tear stuff. So someone mentioned beef jerky. So when you go to like bite some beef jerky to tear off a chunk, where do you tend to bite it? Just slightly off center. You're biting it with your canines and you're biting it with your premolars because they are shaped in such a way that we can kind of get a little bit of shearing, but also great kind of contact so we can tear. Okay. If you look at the premolars of not canine animals, but predatory animals, animals that eat primarily meat, you'll see that they are actually built once again like shears. Okay, so they get very, very good at slicing through meat. Okay. Then we get down to the molars. The molars is where we tend to chew our food. Okay, the molars fit together where we have lots of surface area touching them, and we just jam things in between them until they smash up into the types of pieces that we want them to be in. Okay. Different types of animals, like I said, will have different shapes to their teeth. That's actually one of the ways that we know that Tyrannosaurus Rex ate meat. It's because most of their teeth looks like canines and stuff like that. But there's lots of variation within mammals too. For instance, rats only have one style of tooth all the way back. and they all look like little canines. The same thing goes with marsupials and stuff like that. All right, then we get into the actual muscular tube. So remember, we have our pharynx here. Now, your pharynx has two, or your esophagus has two sphincters. So a sphincter is basically a muscle that is going to allow things to move into or out of the tube. Okay, so we have a superior esophageal sphincter, which is basically going to be right back here at the top of the trachea. That one has to open whenever we swallow. So whenever we swallow, we open that superior esophageal sphincter so that we open up food, open up the esophagus for things to come in. We want it closed most of the time because when you're breathing, we actually don't want all of that air going into our digestive system. Because if air goes into the digestive system, it also has to come out, which means you are either going to burp or you are going to flatulate. That's the only two options once air gets into the digestive system. Okay, we can't just like absorb it because a lot of that air is nitrogen that we don't really deal with very well. Okay, so we typically have this closed. Now we also have the lower esophageal sphincter or inferior esophageal sphincter, which is right down here, and that is what's going to be at the opening of the stomach. So your esophagus basically follows all the way behind, so it's posterior to your trachea, and it's going to go all the way down past your lungs, past your heart, and then finally we're going to enter the stomach inferior to the diaphragm. Okay, so we are now in the abdominal cavity. So whenever you swallow, like I said, you kind of press your tongue up against the top of your mouth, and that's going to force the bolus of food back. So see here, the front of the tongue pushes up, we push the food back, and then we continue pressing the top of our tongue up, and that's going to force the bolus of food back past the epiglottis, and then hopefully down into the open esophageal sphincter, and then down into the esophagus. So then through this rhythmic squeezing called peristalsis, we will push that bolus of food down here into the stomach. And then eventually we will open that lower esophageal sphincter. So that's how the food gets in. Now, once we are in the stomach, some fun things are going to happen. So like I said, we have teeth, we squish everything together, we break things down into bigger pieces or into smaller pieces. But once we get into our stomach, we can start to do some fun stuff. In our stomach, things that stop being recognizable as caramel, peanuts, chocolate, nougat. Instead, we break them down into smaller pieces, into little tiny chunks. Okay, so little tiny pieces that are all mixed in together into a substance that we are going to call chyme. Okay, now that chyme is basically going to be all of the food products, all of the saliva and things like that, but it's also going to be a bunch of hydrochloric acid. Okay, so this is somewhat dilute hydrochloric acid. There's still lots of water, but we're going to have the very acidic contents of the stomach in here as well, but primarily hydrochloric acid. That hydrochloric acid is going to help us do some additional mechanical breakdown of the components of the bite of food that we took in. So it's going to continually help us breaking these things down. But what happens to proteins when you put a protein into a really, really low pH? What do we call it when proteins unfold? What do we call that? Proteins break. Anyone remember what that word is? You're so close. It does start with a D. Denature. Okay. Denature. I could tell you were fishing for it. It's like, I think she knows the word. It's just not coming out right. Okay, so it's denature. That means we're going to take those proteins and unbend them. Bend them into these little lines, which means we can start to break them down. Okay, now we have something else that's going to be happening in our stomach too. One of the other secretions in our stomach is a protein called pepsin. So pepsin is then going to take those really big proteins and break them into smaller protein components. Okay, so the stomach acid does three main functions for us. One, it does continual mechanical breakdown, or continues the mechanical breakdown of our food. Two, it denatures proteins. And three, it also activates pepsin. Because when we make pepsin, it's in this form that we call pepsinogen. And then that stomach acid, that low pH, finalizes pepsin's function, and it becomes the protein pepsin. So it changes pepsinogen into pepsin to allow us to break down the food, okay? And that is all just in the acid of the stomach. We do something else with our stomach too. And we talked about it a second. When your stomach growls, that is because your stomach is squeezing. It is basically like me taking all these pieces of paper in here and squeezing the paper. What am I doing to the pieces of paper in here when I'm squeezing this in lots of different directions? What am I doing to the paper? I'm shifting it around. I'm mixing it. Why am I mixing it? What do I get now? Well, we chewed it so we had more surface area, right? By mixing it around, I'm basically shearing them apart, so continually doing the mechanical breakdown, but I'm also making sure that the stomach acid is touching every single part of it. Okay? So just like if you took a margarita or like if I give you margarita mix and tequila and you just pour the tequila into the margarita mix, what happens? It's not a margarita. You have tequila sitting on top of margarita mix. That's not helpful. Well, I guess it could be. Okay, but that's not a margarita. What do we need to do? We need to mix them together. So what do? You blend them. Okay, that's what we're doing in the stomach. With all that squeezing, that's basically the bartender taking the vodka and the vermouth and then shaking them together. Okay, I'll talk a lot about drinking for someone that doesn't actually drink. Okay, but it's taking the ingredients and it's shaking them together, making sure that they are mixed together. We get continual mixing here. So everything is touching everything else so that we get nice, efficient breakdown of all of the food, okay? Now, your stomach also is gonna be lined with these little folds. We call these gastric folds. So see all these little ridges? These ridges, let's see if I can do this one. All right, so let's, what did this thing just do after I stretched a bunch other than tear? Please don't. Okay, so you stretch the balloon a bunch and then you let it go. See how it forms all these little folds and all these little ridges? So do those little ridges limit my ability to flex it or limit my ability to stretch it? No, they're just evidence that it can stretch, right? That's what these gastric folds are. These are basically raised portions of the epithelial lining. And even some of the muscle will even kind of get into some of these folds a little bit. That basically are going to allow the stomach to stretch. I have personally eaten 1.5, I think it was, what? I think 16 pieces of thick crust pepperoni pizza. I have also eaten 157 shrimp scampi, along with like 3 or 4 rolls and like 2 or 3 like sodas in that front. Okay. Is that how large my stomach is? Is that how large my stomach is right now? Is it big enough to hold a large and a half pizza? Or 150 shrimp? No. So what does my stomach do? It stretches. The shrimp was a bad idea. I got a little bit sick after the shrimp. All that fat, just, I did not feel good. The pizza was actually oddly okay. Shrimp scampi. They're cooked in butter and olive oil. Some of the shrimp may have even been swallowed mostly whole because I was racing my brother. It was Red Lobster. It was the all-you-can-eat shrimp fest. All right, so we can stretch our stomach to allow it to take in more food. Then, after you have mixed all that food up and you push some of it into your small intestines, well, then your stomach can relax and go back to its ordinary shape. Now, if you eat like that a bunch, well, that means your stomach overall will stretch. Okay, so your stomach will be a little larger. If you don't eat big meals like that, your stomach will be overall a little smaller. But that process will take some time. But these gastric folds allow our stomach to have a lot of this flexibility, have a lot of this give and stretchiness to it, so that we can eat a little bit larger than normal meals and stuff like that and be okay, or even just to stretch enough for a normal meal. Because like most of us tend to go 3, 4, 5 hours in between meals. So your stomach will kind of relax and go back to that smaller size, waiting for that next meal to come. Now, in your stomach, we also have 3 layers of muscles. We have a longitudinal layer, a circular layer, and then we have an oblique layer. What this is going to do, this is going to allow us to move and bend, or basically contract the stomach in lots of different directions. And we can do this in different proportions at the same time. So this basically is going to allow us to squeeze it longitudinally, circular, or at a diagonal, all basically at the same time. So this is where we get this nice kind of crazy mixing, is because of these different directions that the muscles can take, okay? Now, we do have a couple of curvatures here. We have what we call the greater curvature, the lesser curvature, and we also have this fundus up here. Now, like I said, we have at the top, we have our lower esophageal sphincter. Someone that has heartburn a lot, so someone with, what's the word I'm looking for? I saw it earlier, so now it's just out of my brain. Chronic heartburn, what do they call it? Yeah, it's fine. Someone that has chronic heartburn, that is a problem because the acid will then back up and move through this particular opening. No, it's actually a symptom of GERD though. Reflux, thank you. Acid reflux, okay? So reflux is basically when this acid from your stomach gets pushed up into your esophagus, and it is no fun, okay? That's what causes heartburn. It's basically acid getting up here and actively interfering with the mucosal lining of your esophagus, which is a problem because you can also start to damage the epithelial tissues, eventually even damage the musculature if it stays that way for too long. So we do that, or we keep it in the stomach by closing this particular sphincter. We also keep the food from entering the small intestine too early by closing this sphincter, which is the pyloric sphincter. So the opening of the stomach is the esophageal opening is the lower esophageal sphincter, and the opening of the stomach into the intestines is the pyloric sphincter. Now, then we enter the first stretch of the small intestines. This is the duodenum. Now, notice, have I said anywhere in this pathway that we are absorbing nutrients? No. So the first parts of the pathways here are not necessarily worried about absorbing the nutrients, okay? Now, you may get a little bit of absorption in your mouth of very simple sugars. That's why if you give someone, say, a Snickers bar whenever they're undergoing, or like orange juice like that, whenever they're undergoing some sort of like diabetic shock or like really low blood sugar, they will get some absorption there, but not very much. Most of the absorption is going to start in your small intestines. Now, the duodenum is going to be this kind of S-shaped curve at the start of your small intestines. Now, there's also going to be a little port right here where we're going to put some additional enzymes in there. Because have we started breaking down proteins yet? So far, actually, I guess we have. So your stomach, we have pepsin, so amylase and lipase, lipids or fats. And then pepsin to start breaking down proteins. So we started breaking them down. But we haven't finished because we are still left with small proteins at this step. So then we are going to get into the small intestine. Now, in the small intestine, we're going to have two organs start to put out some things. And this is going to be our pancreas and our liver-gallbladder. Your pancreas is going to secrete pancreatic amylase, which amylase breaks down what? Sugars. And it's also going to secrete pancreatic lipase, which is going to break down lipids, so lipids, okay? And we're also going to secrete some more proteases, multiple proteases, okay? To finish breaking down those proteins. Finally, so turning proteins into amino acids, okay? Sugars get turned into monosaccharides. Lipids get turned into fatty acids. So we finish chemical digestion in the small intestine. This is also where we're going to start absorption. So we're going to start absorbing these things as we get them broken down. Okay, so we start this process, or we finish chemical digestion and we start absorption in the intestines. Okay, Now, your stomach also has a couple of cool things in it. I forgot that I got a little bit ahead. We're going to call these gastric pits, because on the sides of those gastric folds, or in those gastric folds, we're going to have this special lining of mucosa on the insides of our stomach. And on the insides of those linings, we're going to have these divots, these big holes. We're going to call these holes in that mucosa, we're going to call these gastric pits. That's because we need to secrete the acid, we need to secrete the enzymes. So we're going to secrete those enzymes down here in what we're going to call the gastric glands. Now, we call these gastric glands because they are the glands associating with all of the gastric fluid. Okay, so whether it's additional water, additional mucuses, or additional enzymes, that stuff's all going to come down here from these gastric glands. Now, we have color-coded cells down here because different cells are going to secrete different things. So we have 5 main cells that make up these gastric pits, or at least make up the gastric pits and gastric glands. These are mucus cells, which secrete primarily mucin. Okay, so mucus. We have mucus neck cells, which are going to secrete mucin, but in this case, the mucin that's up here is going to be less acidic. That way, once we get into the glands down here, we have a little bit less acidic environment. Okay, we have parietal cells. That's going to be these ones that are kind of shaped like these little inverted V's. Okay, so these parietal cells are going to secrete our hydrochloric acid. So if I ask you what type of cell secretes hydrochloric acid, it would be a parietal cell. I will ask you on one of the exams, or likely, I know at least one of or two of the questions are asking about it. Parietal cells are what actually secrete our hydrochloric acid. Now, in our stomach, we have pepsin and we have lipase. Okay, so pepsin and lipase are secreted in our stomach as well. So lipase, once again, breaking down lipids. Okay, so you should see that we have lipases kind of everywhere. Okay, taking a lot of fats. Fats are big, takes a long time to break them down. So those are taken care of by our chief cells in our gastric pits. So that's what's coded in purple here. Notice that the nuclei of the chief cells is further away from the lumen. And then finally, we have our G cells. These secrete a protein called gastrin. But these are actually going into the blood, not necessarily into the gastric pit. So there are some slides here talking about those different juices and things like that if you wanted to read that. But that's more physiology than anatomy. So I'm trying to get to the functional anatomy here. All right, so we leave the stomach, and then we're going to have the S-shaped bend of small intestine right here. What did we call that? What do we call this S-shaped bend of small intestine? Yep. Duodenum, duodenum, it doesn't really matter. Like, we don't grade on pronunciation, so pronunciation does not matter in here. And to be honest, I'm not real sure what the right one is. I say duodenum. I have heard people in the medical industry say duodenum, but I've also heard other people say duodenum. So I don't know. Say whichever one in your head helps you spell it. Hooked on phonics, everything. Okay. So here in the duodenum, that is the more common way I've heard it pronounced. Okay. Here in the duodenum, this is where we have our pancreas and our gallbladder secreting their fluids. They're going to secrete it through a common bile duct and then finally into the common duct down here. Okay, that's where we're going to insert those. Now, as we leave the duodenum, we are going to enter this top layer of folds of the small intestine. This top layer of folds is going to be called the jejunum. Okay, so the top set of folds is the jejunum. The bottom set of folds is the ileum. Okay. Top set of folds is the jejunum, bottom set of folds is the ileum. Now, the way that you can remember which one comes last is because the opening of these small intestines into the large intestine is seen right here, and it is labeled ileocecal valve. Ileo. Would this be labeled ileo if that was part of the jejunum? No. Okay, so the ileocecal valve, if you can remember ileocecal valve, that tells you that the ileum is going to be the last part of our small intestine. Now, in the jejunum, we are going to continue doing chemical digestion, but we're also going to start absorption. In the ileum, we are going to finish absorbing all of the nutrients or all of our nutrients, okay? In the large intestine, we are going to absorb water and electrolytes. That is what we are doing in our large intestine, is absorbing water, okay? Let's see if there's a number here. Now, what do you think that we are getting by having all of these folds of intestine, by having this all these convolutions in our intestine? What does that get us? What does that help us do, having all these little folds? I mean, that's true. It does let you do some mixing in some places if we needed to do that, and allow us to move it not too quickly. The not too quickly is actually kind of the important part. It's not that we're moving stuff through it necessarily all that slowly, but quickly is painful. It gives us more time, right? Does it take you less time to drive to Tyler or less time to drive to Dallas? Tyler, because Tyler's closer. It's a shorter distance, right? So this way we can get a whole lot of distance. But the best part is that we get a whole lot of distance in a really small space. By making all these coils, we can shove all these coils into a little tiny space, like right here. That's it. But we can fit 50 or 60 feet of it in here. Okay, so that gives us a lot of room. The more room we have, the more time we have to do all of the both digestion and all of the absorption, which means we get more out of our food. If you look at the intestines of something like, say, a cow, it will be even longer and larger, so they have even more time. Cows and like animals that eat a lot of grasses and things like that have way, way, way more by weight and by size than even you and I do. However, if you look at, say, cow feces that's been dried out on the ground, there is still recognizable grass in it. Why? Because it's not very efficient of a thing to eat. Okay, it's very, very hard to break that stuff down. So they try really, really hard to do it. Cows chew their food twice. They swallow it, get it in their stomach for a little while, and then they throw it back up into their mouth, chew it, and then swallow it again, so that they can have more time to digest it and then hopefully get more absorption once it actually gets down into their gut. Horses have 4 stomachs. Or multiple stomachs. I don't think it's 4, maybe 3. Some ridiculous number. So they move it into each one so they get more chances of doing the digestion part. And then they move it into a really, really long set of tubes to do additional absorption. Still not all that efficient though. Rabbits eat their food twice. They literally eat their food twice. They poop it out and then they eat it again. So whenever you look at like a rabbit cage, there'll be 2 piles of poop. There'll be clean poop and dirty poop. They'll put their first set of poops in one corner and then their second part of poops in the other corner so that they know which one to eat. It's actually not a joke. That's literally what they do. They're weird animals. But it's to give us more time. More time makes us more efficient. We can get more of the nutrients out of our food the longer they stay in our intestine. They also give us lots more surface area to make us even more efficient at doing these things as well. More surface area is very, very important. To help with that surface area, we have even more folds on the insides of the tubes. So our tubes are folded. The insides of the tubes are also folded. Why? So we have more surface area. These also act as kind of like a paintbrush or like a scraper or fingers on a scraper, basically, so you can catch more particulate matter and hold it still for longer. Okay, but we get lots and lots and lots and lots more surface area by doing it this way. So more convolutions means more surface area. We call those, like here, this is one of those folds. So one of those circular folds. We call these circular folds a villus. Okay, so this is an intestinal villus, this little finger right here. Now, these cells are convoluted once again. So if you look at the cells, we form this brush border of these little tiny finger-like projections of plasma membrane or cell membrane to further increase surface area. So we fold the outside of the tube, or we fold the tube itself. We fold the insides of the tubes, all in the name of surface area. Then on those little finger-like folds of villuses, we then put folds on literally the outsides of the cells, all in the name of surface area, so that we get more absorption of the nutrients. That's what this is all about, is getting more absorption of the nutrients. The maximum amount of surface area so that we have a maximum amount of absorption. Now, in these villuses, you're also going to see some specialized lymphatic capillaries. These specialized lymphatic capillaries are what we're going to call a lacteal. So lact, like milk, this is where we're actually going to absorb all of the fats. So any fats that get absorbed from our food are going to move past the blood, like the blood capillaries, and instead they're going to go into the lymphatic system. All right, so how do we get those enzymes into the small intestine? All right, so those enzymes are going to come from the liver up here. So the liver produces bile. Excess bile is stored here in our gallbladder. Gallbladder and liver bile is going to move through what we call the common hepatic duct, okay? So common because it's basically going both to the liver and to the gallbladder. So the common lymphatic duct, or sorry, not lymphatic, hepatic duct, and it's going to come down here to the major duodenal papilla. This is also where the pancreatic duct is going to drain its liquids. So they're going to all go into this primary common bile duct and then finally out into this major duodenal papilla. That's where both enzymes get released at the same time. You block this, you block both bile and pancreatic enzymes all at the same time. Now, bile also does something else that's very, very, very important for us. Bile is what's actually going to allow us to do anything at all with the lipids. Because do lipids play well with water? No. So say you take a bunch of butter and you scrape it onto a skillet. Can you wash that skillet with just water in your sink? Nope. What happens when you put water on it? It just beads up, falls off. So how do we clean the pans? What do we add? Soap. Exactly. We add soap. That soap is going to do what we call emulsification. It's going to allow us to mix the water with the fat, okay? Bile is an emulsifier. It's going to allow us to mix water, which all of our enzymes are dissolved in, with the fat that may be in our food. So it's going to allow us now to get our enzymes access to the big fats so that we can break them into smaller fats. Does that make sense? Okay. So it's going to allow us to mix those two things together. That's why bile is so important, is because it helps us break down the fats. Or it helps us get our enzymes access to the fat. It doesn't actually break them down. Now, the inside of our liver is going to look something like this, where we're going to see a whole bunch of these little hexagons. Hexagons are the bestagons, okay? So hexagons are going to be a series of what we call portal triads, all associated with this single individual sinus here in the middle, okay? Or this hepatic vein. Now, what we're actually doing here, or what we're trying to do here, is we are trying to take all of these little portal vessels here, so these bile ductile, this is where we produce the bile, we're trying to get this into, produce this and then get this into the common bile duct so that we can get rid of it. So this is where we have special little canals for that. But we also have hepatic veins and hepatic arteries. But notice this isn't just hepatic vein. This is a hepatic portal vein. What that tells us is it is carrying blood, not from the liver, it is carrying blood from somewhere else. These hepatic portal veins are connected to blood vessels from all over your abdominal cavity. All of the blood, so all of your now really, really nutrient and stuff-rich blood from your intestines and from your spleen and all that kind of stuff is going to get routed through your liver first before it sends that stuff out to your tissues. That is because your liver is doing something important in all of these little cuboidal cells. Your liver is cleaning that blood. It is making sure, hey, are there broken down bloods? Like, are there old blood cells? Are there bad things in here? Are there toxins in your blood? So when you drink that margarita, you absorb the alcohol from that margarita into the blood. That's why you get the effects of drinking said margarita. But that blood then gets routed into your liver, and your liver is like, hey, ethanol, bad. And it breaks it down. It takes it out of the blood and it breaks it down so that we no longer have it. So what happens after about an hour after drinking your margarita, especially if you've been eating while doing it? Can you tell that you've had a margarita? Nope, it goes away. So over time, as more blood gets routed through your liver, so as that blood keeps going through your liver every time, it gets rid of all those toxins over time. So that's what your liver is doing. So this is how we do it. Basically route that blood in. It kind of mixes with good clean blood from our hepatic arteries. And all of these little cells around those blood vessels are basically taking in all of those toxins that may be in there and helping us break them down. So that then we can return good clean blood into our hepatic veins, and then up through, after the hepatic veins, it goes into the inferior vena cava and back into circulation. Oops, did not realize that was not on. I've been going off of the battery on my computer this whole time. All right, so here we have, hey, where's my pointer? There it is. All right, so here we have one of those little hexagons here. So this is our hepatic lobule here. I'm not going to worry about particular cell shapes or anything like that in here. But if you do see three vessels all associated with one another, one is the bile duct, one is going to be an artery, one will be a vein. That is going to be what we're going to call our portal triad. Okay, so portal triad is going to be the three vessels next to our hepatic nodules. So here's that blood circulation. So blood from the pancreas, the small intestine, the large intestine, all of that blood that could potentially have bad guys in it, and also potentially have bad stuff like alcohols, like other types of poisons and stuff like that, get routed through our liver to help us get it cleaned. And then it can enter the bloodstream again, okay? So that's where we're gonna be dealing with all those toxins and stuff like that. That's also why people that drink a whole bunch will tend to have liver failure or liver issues, because it's the liver that's responsible for getting rid of all of it. What time is it? 11:52. All right, so here we have our pancreas. So everybody remembers the chicken finger. Here's our common bile duct. So pancreas, common bile duct coming down in here. The gallbladder is always, or almost always, going to be coated in green. That's because it is actually green. All right, then we get into the large intestine. So we go through, what did we call this opening? The what? Ileocecal valve. So connecting the ileum to this region of the large intestine, which is called the cecum. The little tail on the cecum is going to be called the appendix. If someone has an appendicitis, that means this thing is inflamed and could potentially burst. If this breaks, what lives in your intestines? Both large and small? Bacteria. More bacterial cells in your gut than you have human cells in your entire body. So that breaks. Now you have blood and all the other kinds of things, which is bad. Lots of bacteria. Now that's real bad. Oh yeah. And remember that feces and all that stuff is really acidic. So now all of that stuff is in here too. That is why if your appendix does burst, you can die. That's why they are so scary and stuff like that. It's simply because it can cause lots of damage in a hurry, because that kind of explodes and all of a sudden you have millions upon millions of bacterial infections trying to start. You have acid coating a bunch of serous membranes, causing damage to all of your other organs and stuff like that. So all really bad, and you're bleeding internally all at the same time. So that's fun. Now, we have a few other regions of our large intestine. We have this one that goes up, so we're going to call this the ascending colon. This one goes side to side, so in the transverse plane, thus transverse colon, descending, and then this one makes a little S-curve. This S-curve is the sigmoid colon. Then we come down here to the rectum, which is basically like the waiting room to leave. And then finally, we have the anal canal, which is where the waste is then expelled. Yes. What causes appendicitis? It could be something like a bacterial infection. It could be other things. I'll be honest, that's not my purview. Bacterial infection is the primary one I can think of. I think it actually said it on one of these previous slides. So here we go. Inflammation of the appendix, mostly due to fecal matter obstructing it. There you go. So fecal matter obstructing it, it gets obstructed. That means the bacteria can't go anywhere, so they can't cause infections at that point. But if it swells, lots of blood supply, lots of bacterial growth, so there's your infection. Then it gets broken, and then we have just general chaos. One fun bit is, though, that we have this T10 referred pain, so around your umbilicus. So if you go into the hospital and saying, I am having excruciating pains, and you point right here, they will take you back to an operating room and take out your appendix. They can do it almost without imaging. They'll image to double check, because they're always in cover your ass mode in hospitals. That's why they do a bunch of tests that aren't actually needed, because they're constantly trying to cover their butts. It's not just to make money, it's to cover their butts just in case they pick the wrong thing. But that pain gets always referred to, it starts in the umbilicus and then it will shift over to the right lower quadrant, and then bam, they know exactly what's happening and they can go in and take care of it. You don't actually need it for anything. So there's your appendix: cecum, ascending, transverse, descending, sigmoid, then rectum, and then finally the anal canal. What we're doing in the large intestines is we are trying to absorb water, and we are trying to absorb electrolytes. The electrolytes are dissolved in the water. So when we absorb the water, we also absorb the electrolytes that are in there. So your sodiums, potassium, salts, all those kinds of things, right? Now, if, say, we don't let the food stay in here long enough, that we don't get very good absorption, and you pass that feces, it will tend to be very, very loose and full of lots of water. Okay, that is also why it's so important when you have diarrhea that you drink a whole bunch of extra water or a bunch of Gatorades and stuff like that. Or that's why your grandma told you to drink Gatorade whenever you had diarrhea, is because we're trying to replace those electrolytes and all that water that we would ordinarily be able to take back in. Does that make sense? Okay. Now, if instead you take out too much water, you'll notice that your poop is going to come out in these little globs, like these little pellets that are going to follow the shape of these little things here. We call these little pouches haustra. So this is why feces that is hard is actually going to have this particular shape, is because it's basically taking the shape on of these haustra, these little pouches. That's also why animals that tend to try and recover as much water as is possible out of their food, because they don't do a whole lot of drinking. This is going to be things like your deer, like rabbits and stuff like that, who drink or eat a lot of water-rich foods. They don't do a whole bunch of extra drinking. They just use the water that's in the food a lot of times. They tend to put out these like really, really dry stools that have this pellet shape. They have that pellet shape because of the shape of these haustra. Horses is another good example. Okay. So it's the shape of these haustra, these pouches, that gives them that shape. Now we also have this little band of connective tissue running down over the top of it. We call this the Taenia coli. So that little line touching here is the Taenia coli. Basically kind of holds everything together. I guess that's about it as far as functional anatomy. That's what things do. Yeah, the rest of the stuff is just physiology at this point. All right. Not to say that that physiology isn't important, but we kind of walked through most of the functional anatomy bits, which is what we're always trying to do in here. So now you have a little bit of time to look at models. We have full size. We have our torso models here on either end. You're welcome to use those. We also have some specialized digestive models back here. So here on the card. There's also some models of jaws. If y'all wanted to look at the various substances. Wisdom teeth are dumb, by the way, because our jaws are not big enough to hold all of those teeth. That is why they tend to cause so many problems, because your jaws simply don't have enough space. So over time, our jaws have shortened. So we just simply don't have the real estate for them. So it causes all kinds of problems. That's why most of us have them removed, or most people will have them removed. You can literally die from some of the issues that those can cause, but it takes a long time to do that. So thus selection isn't efficient. Yeah, you're fine.